So ready to dive into some really cool research today.
Always.
We're talking kidney transplants,
Okay.
but with a twist.
Oh, a twist, all right.
Yeah, imagine a world where blood type wasn't such a big deal anymore for transplants.
I like where this is going.
Yeah, this research is really hinting at that possibility.
It really is, and it's super exciting.
Yeah.
The paper we're looking at today is
Okay.
called Enzymatic Conversion of Human Blood Group O, Kidneys, to Universal Blood Group O.
KG?
It's a mouthful.
Yeah.
It's from Nature Communications, and basically it's all about using special enzymes
Oh, wow.
to actually change the blood type of a donor kidney.
So instead of like searching for that perfect match,
Right.
we could just kind of make any kidney work for anyone.
Yeah, that's the idea.
That would be amazing for like the organ donor shortage and all that.
It would be huge. That's exactly what we're going to unpack today.
Okay.
How they did it, what it means, and what the future might hold. But before we get into all that, let's do a quick blood type refresher.
Okay, back to high school biology.
Exactly.
I remember vaguely ABO, something like that.
Yeah, you got it.
Okay.
To think of it this way,
All right.
your blood cells have these little markers called antigens on their surface.
Okay.
They're like name tags that say A, B, A, B, or O.
Got it.
And then your body also has these things called antibodies. Think of them like security guards.
Okay.
They patrol your blood and make sure only the right blood cells are allowed in.
Okay.
So if you get a transfusion or an organ transplant with the wrong blood type,
Yeah.
those antibodies will attack.
Not good.
No good at all.
But like someone with type A blood,
Mhm.
they have like A name tags on their cells.
Exactly.
And they're security guards.
Yeah,
They're only looking for A, and they'll attack anything else.
you got it.
Okay, that makes sense.
So then what makes type O so special?
Yeah, why is that the universal donor?
Well, type O is kind of sneaky. It doesn't have any A or B name tag. It's like having a blank name tag.
Interesting.
So the security guards don't see it as a threat.
It can slip right past.
Exactly. That's why type O blood can be given to pretty much anyone.
Okay, so that's why people with O blood are always in such high demand.
Yeah, they're the VIPs.
The VIP donors, exactly.
But there's a flip side to that.
What's that?
It also means that people with type O blood
Yeah.
have a much harder time finding a match when they need an organ.
Oh, that's a bummer.
It is, and this paper focuses specifically on kidneys.
Right. Makes sense. Kidneys are super important.
Yeah, absolutely. And there's a huge shortage of donors.
For sure.
In the UK, for example.
Okay.
People with O or B blood types who need a kidney,
Yeah.
they might wait 2 or 3 times longer than
Wow.
people with other blood types.
That's a really long time.
It is a really long time.
So this research wanted to tackle that problem.
Exactly. They wanted to see if they could use these special enzymes to basically remove the A name tags from a donor kidney.
Okay, so make it more like a typo.
Exactly.
And then it could be used for more people.
Yeah, it's a pretty bold idea.
Yeah, it is.
So where do you think they found these enzymes?
I don't know. Maybe they invented them in a lab.
Nope, they actually come from bacteria.
What?
Yeah, scientists are always looking to nature for inspiration.
That's true.
And bacteria, they have some pretty neat tricks up their sleeves.
Like what?
Well, these enzymes in particular, they're like tiny little molecular scissors
Wow.
that can snip away those A antigens.
So they're giving a kidney a blood type makeover.
Exactly.
Yeah.
With bacterial scissors.
That is so wild. Okay, but did it actually work?
That's the big question, right?
Yeah.
And that's where the research gets really interesting.
Okay.
They tested this on real human donor kidneys
Wow.
using 2 different methods.
Okay.
One method was a bit like a regular kidney transplant setup,
Okay.
where they basically perfused the kidney with a special solution at body temperature.
Okay. And the other method?
The other method used colder temperatures.
Which is more like how kidneys are actually stored and transported for transplants.
Makes sense.
So that was important to see if the enzymes would still work in a more realistic setting.
Right. Okay, so they had 2 groups of kidneys.
Yeah.
One treated with the molecular scissors.
Yep.
And then a control group that wasn't.
Yeah, just to compare.
Okay, what happened next?
Well, first they wanted to make sure the enzymes actually did their job.
Of snipping off those A antigens.
Exactly. And get this, after just 2 hours.
2 hours.
Yep, just 2 hours, they saw an 80% reduction
Wow.
in the A antigen expression.
80% in 2 hours. That's incredible.
It's pretty amazing.
Yeah, that's really promising.
It is. But of course, you can't just go chopping up antigens
Right.
and expect everything to be...
Yeah, you got to make sure the kidney is still healthy.
Exactly. That was a major concern.
So did they put the treated kidneys through any kind of test?
They did. They checked for signs of damage
Okay.
and monitored kidney function for the whole experiment. And
Thankfully, the kidneys remained stable and functional.
Okay, good.
No red flags there.
That's a huge relief.
It is so. Both methods, warm and cold,
Yeah.
seem to work without damaging the kidneys.
Amazing. What did they do next?
Well, they wanted to see what would happen
Yeah.
if these treated kidneys were actually transplanted
Oh, okay.
into someone with a different blood type.
Yeah, that makes sense.
Obviously, they couldn't do that with real people.
Right. Too risky.
Way too early for that.
Yeah.
So they created a simulation in the lab.
Oh, so like a test run
Exactly.
before trying it out in the real world.
Hmm.
Clever. Okay. What did the simulation involve?
They took the kidneys that had gone through the cold temperature treatment,
Okay.
and they basically exposed them to a solution
Uh-huh.
containing antibodies against the A blood type.
Oh, okay. So like recreating the scenario
But it's not
where a
just...
type A kidney
Yep.
is transplanted into someone with type B blood.
You got it.
Okay. And that's where those security guard antibodies
Yeah.
would normally attack.
Right.
Okay. What happened with these treated kidneys?
It was remarkable.
Oh, really?
Yeah, the treated kidneys.
The ones that had their name tags snipped off.
Exactly.
Yeah.
They showed very minimal antibody binding.
So it worked.
It's like the antibodies didn't even recognize them as a threat anymore.
Wow. The molecular scissors really worked.
They did. It's pretty amazing.
Yeah, the antibodies were totally fooled.
Yeah, they thought those were type O kidneys.
That's incredible.
Yeah, and to be extra sure it wasn't a fluke,
Mhm.
they also checked the antibody levels in the solution
Okay.
throughout the experiment.
And what happened there?
Well, in the control group.
The untreated kidneys.
Right, the antibody levels dropped.
Because they were latching onto those kidneys.
Exactly like we'd expect.
But in the group with the treated kidneys.
The antibody levels stayed pretty stable,
Oh, wow.
meaning they weren't binding as much.
Okay. That's really convincing.
It is.
It sounds like this enzyme treatment
Yes.
could be a game changer for organ transplantation.
It really could.
But are there any potential downsides
That's a great question.
or risks we need to consider?
Yeah, of course.
What?
Well, one concern is that the effects of the enzyme treatment might not be permanent.
Oh, so like the kidneys, a name tag,
Uh-huh.
it fades for a while,
Yeah,
but then slowly comes back.
that's the worry.
And then wouldn't that trigger rejection eventually?
It's definitely a possibility.
Oh, no.
But there's this interesting thing called accommodation.
Accommodation? What's that?
It's basically the idea that the body can sometimes learn to tolerate a transplanted organ,
Really?
even if it's not a perfect match.
So the immune system just kind of chills out
Yeah, pretty much.
and stops attacking the organ.
Mm-hmm. It's like the security guards get used to those A name tags,
Oh.
and eventually they stop seeing them as a threat.
Interesting.
Yeah, we still don't fully understand how accommodation works.
Yeah.
But it's been observed in some cases of ABO-incompatible transplants.
Oh, wow.
Where people receive an organ with a different blood type.
So maybe there's hope.
Yeah, definitely an area where more research is needed.
Okay. For sure. So accommodation could be the key
It could be.
to making this enzyme treatment really work long term.
Exactly.
Are there any other challenges?
Well, one thing to remember is that this study was relatively small.
Oh, okay.
They only used a few pairs of donor kidneys.
Right.
So we need more research
With more kidneys.
with larger sample sizes to confirm these findings.
Got it. What else?
Also, they only looked at the effects of the treatment for a short period of time.
Oh, okay.
So we need longer-term studies.
Right. To see how the kidneys do over time.
Exactly, like months or even years.
Okay. So more research is definitely needed.
For sure.
But the initial results look really good.
They do.
It feels like we're on the edge of a breakthrough
I think so too.
in organ transplantation.
Absolutely. And this research also raises some big picture questions
What?
about how we allocate organs in the future.
That's a good point. Like, if this technology becomes widely available,
Right.
blood type might not be the main factor anymore
Exactly.
in deciding who gets a kidney transplant.
It would be a huge shift.
Yeah. How would that change the system?
It's hard to say for sure,
Yeah.
but it could potentially level the playing field.
For people with O and B blood types.
Exactly, who currently face the longest wait times.
Yeah. That would be amazing.
It would be huge, a big step towards a more equitable system.
But it also sounds like it could create new challenges, right?
It could.
Like, wouldn't the demand for kidneys skyrocket
Yeah,
if we suddenly remove the blood type barrier?
it probably would.
We already have such a shortage of donors.
Exactly.
Yeah.
That's just one of the many things we need to think about.
The ethical considerations.
Exactly, as this technology moves forward.
Right. We need to make sure it benefits everyone.
Absolutely, and doesn't exacerbate existing inequalities
Right.
in healthcare.
It sounds like there are a lot of complex issues
There are,
to untangle.
for sure.
But it's exciting to think about a future where this treatment could actually become a reality
It is.
and save countless lives.
Yeah, speaking of exciting possibilities.
Yes.
You mentioned something earlier called xenotransplantation.
Oh, yeah. What's that all about?
Xenotransplantation is the idea of transplanting organs from animals into humans.
Whoa.
It sounds a bit sci-fi, I know,
It does.
but there's been some really interesting progress
Oh, really?
in recent years, especially with genetically modified pigs.
Wait, pigs, really?
I know it sounds strange,
Yeah.
but pigs actually have organs
Okay.
that are quite similar in size and function to human organs.
Interesting.
And with genetic engineering, we can modify their genes
Okay.
to make their organs more compatible with our immune system.
So instead of waiting for a human donor,
Right.
we could get a kidney from a pig.
Potentially, yeah.
That seems like it could solve the organ shortage problem.
It could definitely revolutionize the field. But it also comes with its own set of ethical and scientific challenges.
Like what kind of challenges?
Well, there are concerns about animal welfare,
Of course.
the risk of transmitting diseases from animals to humans,
Right. Yeah.
and even some philosophical questions about the boundaries between species.
Wow, that's a whole other can of worms.
It's a complex issue, for sure.
Are there any other cutting-edge areas of research in organ transplantation that you're excited about?
There's another really promising area called bioartificial organs.
What's that?
It involves growing organs in the lab from a patient's own cells.
So instead of relying on donors,
Yeah,
we can just grow organs in a petri dish.
that's the idea.
That sounds like science fiction.
It does, but it's becoming more and more realistic.
Wow.
Scientists have already had some success growing simple tissues like skin and cartilage,
Okay.
and they're working on more complex organs like kidneys and hearts.
Wow, that would be incredible.
It would.
No more waiting lists, no more worries about
Yeah.
rejection.
It would be a whole new era in medicine.
It really feels like we're on the verge of something big.
It's a truly exciting time to be alive.
Yeah, with breakthroughs like this enzyme conversion, xenotransplantation, bioartificial organs.
It's all happening.
It's amazing we're getting closer and closer to a future
Huh.
where organ failure is no longer a death sentence,
Right.
but a treatable condition with options for recovery.
I agree. It's an incredible time to be in this field.
Yeah, it really feels like we're on the cusp of something incredible here
It does.
for people facing organ failure.
Absolutely.
But okay, let's come back to this enzyme treatment for a minute.
Okay.
We've talked about how it works and its potential.
Right.
But what do you think
Yeah.
are the biggest hurdles to actually getting this into hospitals and helping patients?
That's the million-dollar question, isn't it?
It is.
I mean, there are always challenges
Yeah.
in translating research from the lab to the clinic.
Right.
One of the first things we need are larger clinical trials with more patients and longer follow-up periods.
Makes sense. We need to make sure it really works.
Exactly. We need to confirm that this treatment is safe and effective
Yeah.
in a diverse population.
Right, for everyone.
Exactly. And that the benefits outweigh any potential risks.
Okay, so it's one thing to see positive results in a small study,
Yeah.
but it's a whole other ballgame to roll it out to thousands of patients.
It is a whole other ballgame.
What else do we need to figure out?
Well, there's the issue of scalability.
Okay,
Can we produce these enzymes in large enough quantities to meet the demand?
that's a good point.
And what about the cost?
Yeah.
We need to find ways to make this treatment affordable and accessible to everyone who needs it,
Of course.
not just a select few.
Okay, so it sounds like there are a lot of logistical hurdles to overcome,
Definitely.
even if the science itself is solid.
Mm-hmm.
But let's say we do manage to clear all those hurdles.
Okay.
What kind of impact do you think this treatment could have on the world of organ transplantation?
I think it could be absolutely transformative.
In what way?
Well, imagine a future where blood type is no longer a major barrier to getting a kidney transplant.
Yeah,
Wait times could be significantly reduced.
that would be huge.
It would, and more lives could be saved.
Wow.
It would be a massive step towards a more equitable and efficient system.
It's a beautiful vision.
It is.
And I think this research really shows the power of science
Absolutely.
to solve real-world problems
It does.
and improve people's lives.
It's a great reminder that there's always hope,
Yeah.
even in the face of really tough challenges.
So for our listener who's hearing about all this for the first time,
Yeah.
what's the one thing you want them to take away from today's deep dive?
I'd say this. The future of organ transplantation is brighter than ever.
Like that.
With all these brilliant minds working on new solutions,
Yeah.
we're entering a new era where organ failure won't be seen as a death sentence,
Right.
but as a challenge that we can overcome.
That's a powerful message of hope.
It is.
And as we wrap up today, I want to thank you, our listener,
Yes.
for joining us on this journey
It's been fun.
into the world of organ transplantation.
Absolutely.
We hope you found it fascinating
I hope so.
and inspiring as we did.
Me too.
Until next time, stay curious.